Hello, my name is Nand and welcome to my poster. This poster is about a new multi-omics approach for gene prioritization. Now, gene prioritization is key in locating genes of interest in ever-increasing datasets. Currently, there are already various multi-omics integration tools available for this purpose. However, they're not always applicable or they suffer from some limitations. For example, there are several tools that can only handle networks containing genes. Here, I'll present MONIR, multi-omics network impact-based ranking, which is a general-purpose gene prioritization tool that ranks genes based on the number and proximity of omics changes in a network. It consists of 3 modules. The first one is for network construction, the second and main module is for gene prioritization, and then you have a third module that helps you create subnetworks for downstream visualization. To showcase our tool, we reused in-house transcriptomics and metabolomics data concerning the infection of Pseudomonas aeruginosa by bacteriophage Lus19 in order to find genes with a potential regulatory role during the infection process. To start our analysis, we first need to make a network, and that's what's shown in next to number 1. So here I used the first module of MONIR to create a network that contains all information from the KEGH and Regulome-PA databases. This includes protein-protein, gene expression, protein compound, and metabolic interactions. In total, our network covers around 1,250 compounds, which are the green nodes, and around 3,500 genes, which are the light blue colored nodes. That's around 61% of the Pseudomonas aeruginosa genome. In the second box, I visualize the input data. We possess both transcriptomics and metabolomics data, and specifically this data for 3 time points during infection. That is 5, 10, and 15 minutes after infection. In the second and third column, you can also see the number of differentially expressed genes and the number of significantly changing metabolites per time point. In total, we start with around 626 unique differentially expressed genes across 3 time points. Next, we use both our input data and the network to rank all those differentially expressed genes. And that's covered in box 3. So all differentially expressed genes are ranked based on the number and the proximity of omics changes in the network. To help you better understand the proximity, what I mean by proximity, I have created a small network next to this box. Here, proximity is visualized by the saturation of the red color. Next, the omics changes or the nodes that significantly change in either metabolite concentrations or gene expression are visualized with the green outline. So in our tool, proximity is determined using a diffusion-like approach starting from the gene of interest, which is the yellow one. So you can see that the color red is much more intense next to the yellow node, so they're closer, and it is much fainter when you move away from the yellow node in the network. The unique thing about our algorithm is that to calculate this proximity, also the missingness of data, the directionality, and the type of interaction are all taken into account. Now, we calculated, based on this proximity and the number, we calculated an impact value for all those for all those differentially expressed genes, and then we filtered them to only retain very high scoring genes. The results of that are given in the figure below, so next to number 4. What you can see here is that the high scoring genes are mainly involved in signal transduction carbohydrate and amino acid metabolism. But what you can also see all the way on top is that there are a few genes that are involved with drug resistance. That can be very interesting. We visualized those genes that are involved with that process in a subnetwork, and that you can see next to number 5. This subnetwork visualization reveals that those two genes, which are FOP and FOQ and encode a two-component system, are downregulated and also seem to be downregulating downregulating genes from the ARN operon. This operon is involved in LPS modification and polymyxin resistance. So, in other words, our analysis seemed to suggest that there is a possible synergistic effect between polymyxin and Lus19. To conclude, starting from our 626 unique differentially expressed genes, Monir allowed us to home in on 43 genes of interest. These genes are involved in biological processes such as signal transduction, carbohydrate and amino acid metabolism, and polymyxin resistance, and the latter could hint at a potential synergistic effect between Lus19 infection and polymyxin in combating Pseudomonas aeruginosa. So, our analysis shows that Monir can help to identify genes of interest and potentially reduce the number of time and labor-intensive experiments. Thank you for your attention. Goodbye.